Bismillahirrahmanirrahim. Dear students, we are recording lecture series from IGCSE and O level biology. This is written by me according to the latest syllabus of IGCSE course code 0610 and O level biology course code 5090. It's according to the latest syllabus uh, 2023 to 2025 syllabus, and it's complete, almost complete uh, course, which is very simplified, and it's available on student resource. Dear students, we have recorded two lectures uh, from the series. Today we are going to start the third lecture. Uh, Here, my number is also given on the screen, so you can contact if you want to have any discussion or any problem related to this one. Okay, dear students, in the first chapter, we have studied characteristics and classification of living organisms, and it was about 25 minutes video or 25 minutes lecture. The second lecture was about 7 minutes, which was about the cell structure and cell organelles. And now it's the third lecture, which is uh, the similarities and differences between animal and plant cells. Dear students, we have already studied about cell theory, that is, all the living organisms are made up of cells, whether they are animals or plants, so they are made up of cells. And all the cells are produced from the pre-existing cells, and cell is the basic structural and functional unit of the life. Now, if we talk about the similarities between animal and plant cells, then what are the similarities? That is, cell membrane, cytoplasm, nucleus, vacuole, mitochondria, uh, ribosomes, and endoplasmic reticulum may be smooth and rough, so they are the common which are found in both the animal and plant cells. Now, if we talk about the differences, so then these are few differences. For example, size, shape, here some other organelles, like if we talk about the differences between animal and plant cells, the plant cell is large and has a fixed rectangular shape, whereas animal cell is small and irregular or round in shape. Cell wall is present in plant cell, but cell wall is absent in the animal cell. The nucleus lies on one side of the plant cell because of the big vacuole in the center, and the nucleus lies in the center of the animal cell. Mitochondria are present in fewer number in plant cells, and mitochondria are present in the large number of the animal cell. The reason is this: plant cells need less amount of energy for its activities, and the animal cells need comparatively more amount of energy. So that's why the plant cells have less mitochondria, and the animal cell have more number of mitochondria. Plastids are present in the plant cells, and these plastids contain chlorophyll, and chlorophyll is used for photosynthesis. So the plastids are absent in the animal cell. Next are centrosomes. These are absent in the plant cells, but the centrosomes are present in uh, animal cell, and one large central vacuole is present in plant cells, but many small vacuoles are present in the plant cell. Next, my dear students, are a bacterial cell. You know, in this chapter, we have to study about the prokaryotic and eukaryotic cell. So you can say that a bacterial cell is a uh, prokaryotic cell. Its diagram is given on the screen. You can see it contains uh, it contains plasma membrane. Over the plasma membrane and cell wall are the pili. Next, it contains nucleus without nuclear membrane. So this is called as nucleoid. You can say simple DNA without nuclear membrane. Then its capsule, which is around the cell wall, and next is the cytoplasm, and within the cytoplasm, a circular DNA is present, which is called as plasmid, and the bacteria also contain flagella, which are used for movement of the bacterial cell. And pili are the small structures with the help of which it can be attached with the other uh, bacterial cells for the purpose of conjugation. But we will study later on. Okay, dear students, a bacterial cell. Bacteria are microbes with a cell structure simpler than that of many other organisms. Uh, their control center, containing the genetic information, is contained in the single loop of DNA, and some bacteria have an extra circle of genetic material called a plasmid rather than a nucleus. Uh, Next, dear students, here are two pictures which you can compare a prokaryotic cell and a eukaryotic cell. So on the right is a eukaryotic cell and on the left is a prokaryotic cell. So here you can see uh, the bacterial cell is smaller in size. It contains flagella, pili, plasmids, nucleoid, cell wall, capsule, and some things common, which are uh, cytoplasm, ribosomes, and plasma membrane. So these are the commonalities. Now let's talk about the differences. So the eukaryotic cell contains uh, endoplasmic reticulum, both rough and smooth, which are membrane-bound organelles. Similarly, it contains uh, mitochondria, vacuole, Golgi apparatus, uh, intermediate filaments, nuclear envelope, chromatin, DNA, nucleoplasm, nucleus, nucleolus, and microtubules, lysosomes, microfilament, and peroxisome, etc. So these are the larger cells with more organelles, which are mostly membrane-bound organelles. Next are the sizes of the specimens. First of all, we have to study about the magnification. It is the observed size of an image divided by the actual size of the image. So magnification's formula is magnification is equal to image size over actual size. And if you want to study the actual size, it will be equal to image size over magnification. So there are uh, 10 lakh micrometers in 1 meter, and 10,000 micrometers in a centimeter, and 1,000 micrometers in a millimeter. Uh, so 1 micrometer is a 1,000th of a millimeter. Next, my dear students, which we have to study in this chapter are different types of the cells, which are root hair cells, red blood cells, uh, xylem cell, muscle cell, nerve cell. Uh, Next are the bacterial cell and ciliated cell. Similarly, goblet cells. Next are muscle cell, palisade mesophyll cell, red blood cells, sperm cell, egg cell, and then we have to study about the cell organization. Different cell organelles combine together to make cells. Cells combine together to make tissues. Tissues combine together to make organs. Organs combine together to make uh, system, organ system, and systems combine together to make an organism. So mostly multicellular organisms contain many systems. Like humans, we have different systems like digestive system, circulatory system, excretory system, nervous system, endocrine system, etc. Next are the uh, This line can represent this in a better way. Cells combine together to make tissue, tissues combine together to make organs, organs combine together to make organ system, and thank you very much. Organ systems, organ systems combine together to make humans, like we are the humans. Uh, Next, my dear students, how we can observe animal and plant cells, especially the plant cells under microscope. So they can be observed under microscope in such a way. First of all, take a clean glass slide, then take about 1 centimeter square of onion epidermal layer, which should be spread on a glass slide. Then put iodine solution over that to make it colored or for staining. Then put a cover slip over this with the help of a mounted needle. Then press it gently and wipe out the surplus material with the help of a tissue paper or blotting paper. Then uh, observe the slide under microscope, firstly at low power and then at high power. And then you can see the cells, which will be the brick-like structures, and they can be easily observed. And if you want to observe the animal cell, you may take a piece of chicken liver, crush it on a center of glass slide and put a drop of methylene blue stain or maybe iodine solution on the top of the smear. Then cover it with a cover slip. Remove the extra material with the tissue paper and observe the slide under microscope, firstly at low power and then at high power. Then uh, certain rounded structures can be observed under microscope. So this is the practical work. At this time, we can't show you the pictures, but later on can be shown to you. Okay, dear students, best of luck. We have completed this chapter.